Hello and hello everyone, welcome to class 12 biology. In our previous video on immunity, we discussed about the two types of immunity, innate immunity and acquired immunity. In innate immunity, we discussed about different types of barriers, physical barriers, physiological barriers, cellular barriers and cytokine barriers. And then in acquired immunity, we talked about those two special lymphocytes, B lymphocytes and T lymphocytes, and their role in acquired immunity. And then I told you that B lymphocytes, they produce antibodies, right? And the response, immune response in which the antibodies are involved, it is called as humoral immune response. And there is another type of immune response called cell-mediated immune response in which the T lymphocytes are involved. Right. So we studied about those two types of immunity. They are innate and acquired immunity. In this particular video on immunity, we will talk about vaccination and immunization. We will talk about some basic concepts regarding vaccination and immunization. And later on, we will talk about two non-infectious human diseases, allergies and autoimmunity, which has got something to do with immune system. Right. And then at the end, we will talk about the immune system, human immune system. What are the components of human immune system? So let us discuss about vaccination and immunization. By looking at these two terms, it seems as if these two words can be used interchangeably. Right? But let us try to understand the difference between the two by looking at the definition of immunization. Immunization is the process whereby a person is made immune or resistant to an infectious disease, typically by administration of a vaccine. Right. So by administering vaccine, by vaccination, what we are trying to achieve is we are trying to make a person resistant to a particular disease. Right. And that particular process is called as immunization. Right. So we can take an example of the pulse polio program. We all know about pulse polio program in which the infants who are less than 5 years of age are administered pulse polio drops. The pulse polio drops are nothing but vaccine. Right. So that particular vaccine is administered in order to make those children resistant or immune towards polio virus. Right. And the entire process is called as immunization. So you can note it down, the definition of immunization, in order to understand the difference between vaccination and immunization. But still, it can be used interchangeably. Okay. Now, the principle of vaccination is based upon the property of memory of the immune system. I told you in our previous video that our acquired immune system has got two important properties. They are, first, specificity towards a pathogen, and then second one is memory. Right? I told you our acquired immune system can keep the information of a particular pathogen that they have come across in their memory so that they can show a stronger response when they come across the same pathogen later on. Okay, so the principle of vaccination is based upon the property of memory of the immune system. And the vaccine that we take, whether let it be polio drops or chickenpox vaccine or any vaccine, the vaccine contains antigenic proteins of the pathogen, or it contains the inactivated pathogens. Antigenic proteins means, last time I told you, if we have a pathogenic bacteria or a virus, if we take the proteins present on the surface of the bacteria or virus, those proteins, they form the antigens, right? And we can use those antigens in preparation of a vaccine for that particular pathogen. Or we can take the entire pathogen, make it inactive. Inactive means now this particular pathogen can no longer cause disease, right? Such inactivated pathogens can be used in a vaccine. Okay, so vaccine contains antigenic proteins of pathogen or inactivated pathogens themselves. So nowadays what we are using is we are using recombinant DNA technology or biotechnology to produce vaccines on a much larger scale. We don't have to grow virus and bacteria in order to make vaccines. We can use recombinant DNA technology to produce vaccines on a larger scale. Right? And example for that particular processing is we have prepared hepatitis B vaccine from yeast. Right? We have produced hepatitis B vaccine from yeast by using recombinant DNA technology. We will be studying about the recombinant DNA technology in detail in our later chapters. Right now, we will try to get the general idea about how hepatitis B vaccine was created by using biotechnology or recombinant DNA technology from the yeast cells. So a yeast cell over here, a yeast cell on its own cannot produce hepatitis B vaccine because a yeast cell cannot produce hepatitis B viruses antigens. Right. So what we need initially is we need the genetic material of the hepatitis B virus. So from that particular genetic material, we isolate a particular gene which is responsible for producing hepatitis B antigen. First, we isolate the DNA. We isolate a particular segment of DNA which is responsible for producing hepatitis B antigen. And then we insert that particular gene into the plasmid of a bacteria. Plasmid of a bacteria is a vector. It can carry that particular gene into the yeast cell. So we create a recombinant DNA, which is the combination of plasmid along with the gene of interest, and then we insert that recombinant DNA into the yeast cell. Now the yeast cell has got the genetic information to produce hepatitis B vaccine or hepatitis B antigen. Right. So this particular yeast cell, when it is allowed to grow in a medium, in a large tank, it can now produce lots and lots of hepatitis B antigen. Right. And that particular antigen can be used for the production of hepatitis B vaccine. So this is the process of recombinant DNA technology, which is used in the production of hepatitis B vaccine. I hope it is clear, right? You don't have to study this in detail. We will be studying about the recombinant DNA technology in our biotechnology unit. Now there are two types of immunization: active immunization and passive immunization. So in active immunization, we administer a person with vaccine. Right. And the person's own immune system will start producing antibodies against a particular pathogen, thereby becoming resistant to that particular disease. That is active immunization. In passive immunization, what we do is we administer a person with preformed antibodies. Right. So the immunization with preformed antibodies for quick action is called passive immunization. So you can see a quick action is required, a quick immune response is required. For such conditions, we administer passive immunization. What are such conditions under which a quick immune response might be required? In your textbook, two examples are given. They are tetanus and snake bites. In the first case, when a person gets a deep wound, there is a risk of bacterial infection, bacteria called Clostridium tetani, which can cause a very deadly bacterial infection disease called as tetanus or lockjaw. Right. So that can be quite fatal. So in such situations, a preformed antibodies against the bacteria or a preformed antitoxin against the bacterial toxin is administered to a person for a quick immune response. Right. So that is passive immunization. In the second case also, when a person is bitten by a venomous snake, right, the venom can quickly kill a person. Right. There is no time for the person's immune system to start producing antibodies against it. Right. So in such situation, again the hospital will administer preformed antibodies against the snake venom. In both this situation, passive immunization is administered. In this diagram, we can see the process of making antivenin against snake venom. In the first step, the snake venom is collected. This is called as milking the snake. The snake venom is injected into animals like horses and pigs. So the snake venom is injected in increasing doses so that these animals, they don't die and their immune system starts producing antibodies against the snake venom. And when these animals, they become immune to snake venom, their blood is collected and the antibodies are purified. And those antibodies, they are sold as antivenin in the market. Such antivenin, they are administered as passive immunization against people who are bitten by venomous snakes. So that was about vaccination and immunization. And now next we will discuss about allergies and autoimmunity. So these two are non-infectious diseases which are related to the functioning of our immune system. Allergies or allergic reactions are diseases which happens due to abnormal functioning of your immune system. Right. So the exaggerated response of the immune system to certain antigens present in the environment is called allergy. By exaggerated, we mean unreasonable response. right? Exaggerated response means some unreasonable response of your immune system towards certain antigens, certain substances which are present in the environment, right? And these antigens or these substances, they are not necessarily pathogenic or toxic to your body, but your immune system, it shows some abnormal response or exaggerated response towards that particular substance, and that particular reaction is called as allergy. Allergy can be triggered by substances like mites in dust, pollen grains, animal dander, or even it can be some food materials, right? So these substances which can trigger allergy are called as allergens. Okay, and we can call allergy as hypersensitivity. I told you, right, these substances they are not potentially pathogenic or toxic to our body, but our immune system, it responds in a exaggerated manner, right? So that is why it can also be called as hypersensitivity towards these chemicals. Now, the symptoms of allergic reaction include sneezing, watery eyes, runny nose, and sometimes difficulty in breathing. And these allergic reactions are caused due to your body producing immunoglobulin E type of antibodies, right, against these allergens, and your mast cells, your body's mast cells, they produce histamine and serotonin when it comes across allergens, right? Histamine and serotonin, they are inflammatory chemicals within your body, right? So they causes inflammations. And in order to control allergy, you have to avoid first allergens. You have to avoid allergens. In order to know your allergens, the doctors will perform diagnosis in which the doctor will test different allergens in small doses, and then they will observe the reaction. So if you show allergic reaction towards certain substances, and you will be told that you are allergic to such and such allergens. Okay, if you suffer from allergic reaction accidentally, you will be administered antihistamine, adrenaline, and steroid as a treatment measure. So regarding allergies, one fun fact is given in your textbook. Not so much fun for people suffering from allergies and asthma, but this fact is given in your textbook, saying that more and more children in metro cities of India suffer from allergies and asthma. And in previous board exam questions, they used to ask why more and more children in metro cities of India suffer from allergies and asthma. Right? So the answer given in your textbook says that due to very protective environment in which these children grow in metro cities, right, this makes them prone to suffer from allergies and asthma towards many different substances, right? Since these children, they grow in a room where there is air conditioning, right? They don't go out to play much, right? They don't touch pet animals, they don't have pet animals, right? They don't go out and play in the mud. So they grow in a very protective environment, and therefore they do not come in contact with many different substances in their early life. So therefore in their latest life, they become allergic to those substances, right? And research also says that the early exposure of a children to certain substances makes them less prone to developing allergic reaction towards those substances, right? So you can write the answer in somewhere in those lines. So the next human disease which is caused due to abnormal functioning of immune system is autoimmunity. Remember, I told you that our body's immune system has got one very important property in which our immune system can differentiate between body's self cell and non-self foreign pathogens, right? Now what happens if our body cannot differentiate between the two and starts attacking our own body cells, right? In such situation, it can lead to a disease called as autoimmune disease, and the condition is called as autoimmunity. So when the body attacks its own healthy normal cells, it leads to autoimmune disease. Now, what causes our body's immune system to start attacking our own healthy cells, right? This happens due to genetic reasons, right? And other reasons are unknown, right? So genetic reason, because if there is a tendency that if a person is suffering from autoimmune disease, right, he or she can pass the same disease to his or her children. So that is why autoimmunity seems as if it is genetic, right? It has got genetic basis of inheritance, and other reasons are unknown, right? And example given in your textbook of one autoimmune disease is rheumatoid arthritis. This is very important. You have to remember this particular name. Rheumatoid arthritis is an example of autoimmune disease in which the body's own immune system starts attacking the joints, which includes bones and cartilages, which leads to warm, swollen, and painful joints. So rheumatoid arthritis, it affects our digits, wrists, and knee joints. Okay, so it leads to very painful conditions, and it can also deform your digits. It can lead to swelling and deformation. Look how the fingers get crooked. Okay, so rheumatoid arthritis is an example of autoimmune disease in which the body's own immune system starts attacking the body's own normal healthy cells. Okay, so I hope you're clear regarding autoimmunity. Now let us discuss about the human immune system. Right, so the human immune system consists of lymphoid organs, lymphoid tissues, cells, and antibodies. So we have already studied about antibodies, right, which is a Y-shaped protein which is synthesized by B lymphocytes. Okay, so we have also studied about different types of cells which are involved in both innate and acquired immunity, right? So those are the cells of the immune system over here. They all originate from hematopoietic stem cells and then differentiates into different types of functional immune cells, right? We have memory B cells, plasma cells, or B lymphocytes. Then we have T cells, cytotoxic T cells, helper T cells. We have mast cells, neutrophils, eosinophils, dendritic cells, macrophages. All these cells, they are part of our immune system because they have got protective role. They protect us from different types of antigens, different types of pathogens. Okay, so we have discussed about them. Now the only thing left to discuss about is lymphoid organs and lymphoid tissues. In our body's immune system, lymphoid organs can be categorized into two groups: primary lymphoid organs and secondary lymphoid organs. Under primary lymphoid organs, we have bone marrow and thymus. And under secondary lymphoid organs, we have spleen, lymph nodes, tonsils, Peyer's patches, and our appendix. Right. So these lymphoid organs, they serve as a site where different types of lymphocytes like B lymphocytes and T lymphocytes, they can differentiate and mature into functional lymphocytes. And they also serve as a site where these lymphocytes, they can come in contact with various foreign antigens and where they can launch their immune response. If you look at the diagram of human lymphatic system over here, we have thymus gland and bone marrow, which are the primary lymphoid organs. So within the thymus and bone marrow, the immature lymphocytes, they differentiate into antigen sensitive lymphocytes within these two primary lymphoid organs. Right. So immature lymphocytes, they become functional antigen sensitive lymphocytes like B lymphocytes and T lymphocytes within these primary lymphoid organs. And the fun fact, T lymphocytes, they mature within the thymus. B lymphocytes, they differentiate and mature within the bone marrow. So you can correlate between their names and their site of differentiation. In secondary lymphoid organs, we have spleen over here. We have lots of lymph nodes. We have tonsils. We have Peyer's patches in the small intestine. Then we have appendix. Right. So these are the secondary lymphoid organs. So the secondary lymphoid organs serves as a site for interaction of lymphocytes with the antigens. When a pathogen enters your body, the pathogen themselves or the antigens of the pathogen reaches these secondary lymphoid organs, and inside these secondary lymphoid organs, the lymphocytes are there. Therefore, the lymphocytes and the antigens or the pathogens, they interact with each other, and the lymphocyte, they proliferate within these organs and they launch their immune response. And this leads to the inflammation of these secondary lymphoid organs. Right. And if you remember, when you suffer from sore throat, the doctor checks and looks within your mouth with the torch, right? So what he or she is looking for within your mouth is he or she is looking for signs of inflammation of your tonsils. So we have here pharyngeal tonsils or adenoid tonsils. We have palatine tonsils over here. So these are secondary lymphoid organs where the interaction between the pathogen which is causing the sore throat and your lymphocytes are taking place. So this interaction could lead to inflammation, right? When the lymphocytes starts fighting with the pathogens, right? Or when lymphocytes, they react with the antigens, right? What happens? Inflammation happens. So the doctor will look within your mouth and see whether inflammation has occurred or not. If inflammation is there, it shows the sign of infection. Now we have these information regarding specific lymphoid organs which are given in your textbook and which are important from examination point of view. So the first one is about bone marrow. So bone marrow is over here. So the bone marrow is said to be the main lymphoid organ. Okay, all the blood cells are produced inside the bone marrow. RBC, WBC, platelets, and all those blood cells, they are produced within the bone marrow. Okay, so then next we have thymus. Thymus is a bilobed organ, means there are two lobes, right? Thymus is a bilobed organ located beneath our breastbone or sternum near to our heart. It is large at the time of birth, and by puberty it reduces to a very small size. It shrinks as we grow older. Okay, so thymus, this is one peculiar information about thymus you should remember. It is large at the time of birth, and by the time you reach puberty it has reduced to a very small size. Okay, that is about thymus, which is also a lymphoid organ, primary lymphoid organ. Then we have spleen. So spleen is located over here. Spleen is a large bean-shaped organ which acts as a filter of the blood by trapping blood-borne microorganisms. So if the microorganisms they are found within the blood, spleen, it acts as a filter, right? It cleans those microorganisms away from the blood. And the spleen also contains lymphocytes and phagocytes, which helps in cleaning process. Okay, and spleen also acts as a reservoir of erythrocytes, means it stores lots and lots of RBCs within itself. Okay, so it acts as a reservoir of erythrocytes. And then we have lymph nodes. So all these yellow dots over here, they are all lymph nodes. These are small solid structures. Lymph nodes, these are small solid structures located at different locations within our body. And what these lymph nodes do is they trap the microorganisms or other antigens within them, and they provide a site for the interaction of lymphocytes with those antigens. And in these locations, the lymphocytes, they get activated. And finally, we have MALT, short for mucosal associated lymphoid tissue. So this is a lymphoid tissue. So within the linings of the major tracts, respiratory tract and digestive tract, then we have urogenital tracts, right? So mucosal associated lymphoid tissues are found within those major tracts. Why? Because our respiratory tract, our digestive tract, our urogenital tracts, those are all exposed to foreign pathogens present in the outside environment, right? So since they are exposed to the outside environment and those foreign pathogens, they constantly come in contact with those pathogens. So therefore, a defense mechanism should be ready over there as well, right? So therefore, we have those mucosal associated lymphoid tissues. For example, in the small intestine, we have these Peyer's patches. So these are the Peyer's patches which are present in the mucosal layer of the small intestine, right? So these Peyer's patches, they contain lots and lots of lymphocytes and macrophages and other immune cells, right? So whenever a pathogen comes from the outside through food and water, right, they have to go inside, and when they come in contact with these Peyer's patches, the immune cells which are present within the Peyer's patch, they launch immune response and protects us against those pathogens. Okay, so these are called as mucosal associated lymphoid tissue. Peyer's patches in the small intestine is an example of mucosal associated lymphoid tissue. So that is all about the immune system or immunology. Now in the next video we will discuss about HIV/AIDS. Okay, so please read about HIV/AIDS and then you can watch the next video. Thank you.